So I have the great pleasure tonight to introduce our speaker, Dr. Aditee Mitra, who is a botanist, ecologist, and marine plankton specialist. And her background allows her to interface with a really wide science base in both research and teaching. And she also is a very accomplished presenter of outreach events in a wide variety of circumstances. Her research basically has been to look at marine plankton, and she's been a key driver of a new, what might call planktonic-centric view of the oceans, thinking about those organisms that contribute 50% of the oxygen that we breathe. She has a stellar CV, and she has very interesting prizes that she has been awarded, one of which is the Yuva Ratan Award for last year, which is presented at the 39th International Convention of Non-Resident Indians. And this is something that's an extremely important award, which is given to Indians who are no longer resident in the country. And I think it's a huge mark of esteem that you've won that. I think it's absolutely fantastic. She's a visiting professor at the Free University of Brussels, and she's been a media host on BBC Countryfile. So I'm really delighted to welcome Aditee here tonight to talk to us about mixoplankton. And I think I kind of know what the answer is whether they're mini monsters or perfect beasts, but we'll find out. Thank you so much, Aditee, and over to you.
Thank you very much, Sandy. That's a very kind introduction. Thank you very much to Professor Diane Edwards, to the Royal Society of Biology South Wales branch and the Linnean Society for inviting me for this very prestigious event. I'm really honored to be here. Thank you. So, so on with the talk then. So I would like to start with thanking all my colleagues. So the work I'm going to present today is work which we have been doing over the last decade and a bit more, and our outputs from various grants. So, so thank you to everyone for getting us where we are today. So I'll start with what are plankton. These are organisms that can drift in the oceans but cannot swim against water currents of tides. So you get plankton of all sorts. But majority of these plankton, the vast bulk of the marine life, comprises of microbial plankton. This is both numerically and in biomass. So microbial plankton, if you're wondering what they are single-celled organisms. And within this, we can again very broadly divide them into three. We have got the heterotrophic bacteria. These are then we've got the photosynthesizing cyanobacteria. Both the bacteria and cyanobacteria fall within the prokaryote group. And then we have got the single-celled protists. The protists are like us, eukaryotes. So we fall in the same group when you look at the broader picture. So why plankton? Why microbial plankton? At the very beginning, when we had early Earth, it was very much like Venus. The atmosphere was full of carbon dioxide, more carbon dioxide than oxygen. Over millennia, it was the microbial plankton which changed this atmospheric carbon dioxide to atmospheric oxygen and made the Earth as it is today. So we live on Earth, or the planet is as it is because of microbial plankton. And in today's world, microbial plankton still play a very important role. They are the main drivers of life in the oceans. So we have got the microbial plankton in the ocean at the base of the food web, which make food, which are eaten by the mesoplankton and the fish, etc. And the nutrients from the higher trophic levels then go back to support the microbial plankton. So what we can see in the ocean life when we look at it on the food web, it's very much like our land food webs with a very clear plant and animal dichotomy. So this is what we're going to look at is the dichotomy and the microbial plankton and where the mixoplankton fit in this. So the protist plankton dichotomy, we start with the at the bottom end with the food producers, like on land, we've got a plant-like phytoplankton. So these phytoplankton will use carbon dioxide, light, and nutrients to make food, and they release oxygen and different dissolved organics like sugars. And then at the other side, we have got the animal-like zooplankton, the protozooplankton, which are eating prey, taking up oxygen, releasing carbon dioxide, nutrients, and also defecating waste. So when we look at this very broadly, the plankton dichotomy, we got at one end the phytoplankton, at the other side the protozooplankton. And the question then is, okay, so which one came first in evolutionary terms? Was it the plant-like phytoplankton or the animal-like protozooplankton? When I first looked at it, I would have thought originally it would be the plants. The plants has to come before the animals, but actually not in protist world. It was the protozooplankton which came first. So they were their ancestral form, and the phytoplankton are more recent forms. And then how did this happen? How do we get a plant from a plant-like organism from an animal-like organism? So this is one of the thoughts which is there about how this happens. We've got the protozooplankton, which is eating, etc. There was in early Earth, there would have been cyanobacteria. So these are the photosynthesizing prokaryotes. They would have been taking up. The protozooplankton would have been ingesting the cyanobacteria and digesting and growing and replicating. And at some point, they would have kept the cyanobacteria, the plastids, the photosynthesizing material from the cyanobacteria, and gradually started replicating it, and eventually got the capability to have the genetic code to have the chloroplasts and plastids and have it themselves. So basically, they would have evolved to become a photosynthesizing phytoplankton. So that's all very well. But then the question is, okay, so what happened? How did this happen? It's how did it go from a protozooplankton to a phytoplankton? It all makes sense, but did it just happen in one step or two steps or how many steps? And the answer here is the mixoplankton. So that's where the clue is of how we got from the ancestral form to the more recent phytoplankton. So what are mixoplankton? So mixoplankton are single-celled planktonic protists, so they're eukaryotes. They have the ability to acquire nutrition through phototrophy and through phagotrophy. So by photosynthesizing and by eating, they can get their nutritional requirements. Are there new discoveries? So this is something new? The answer is no. The popular perception, though, was we've got the phytoplankton, which are the primary producers. Then we've got the protozooplankton, which are the primary consumers, eating the phytoplankton. Then the copepods eat these fishes, these, etc. And somewhere in between, we have the mixotrophic protists, doing both a bit of photosynthesizing and a bit of eating. And what people believed till, well, for decades, they were really not in mainstream research because they thought they were inferior, because they were not doing one of the making food or eating food purely. Therefore, they were mixing strategies, so they were inferior. And some people call them freaks. Politely, some people call them curiosities. So before we go further, you probably notice I've used two different terms, mixotropes versus mixoplankton, mixotrophic. And why have we come up with the term mixoplankton? Because this is a very new term, which we suggested in 2019, proposed in 2019, instead of mixotrophic protists. And this is the reason why. So the definition of mixotroph is an organism which can do autotrophy and heterotrophy. So and then we get mixotrophy. So basically, autotrophy is that it's making food. Heterotrophy, very simply, is eating food, and mixotrophy is they're doing both. Now, when we have in the marine food webs, when we look at the mixotrophy, we get phototrophy, a type of autotrophy. So organisms are using light, dissolved inorganics, so carbon dioxide, nitrate, ammonia, etc., to make food. And then we have got osmotrophy, which is a type of heterotrophy. So this is taking up dissolved organics like sugars, etc., amino acids, etc., to again grow. So when we have this phototrophy and osmotrophy, we get mixotrophy. And when we look at the planktonic, microbial plankton, we find that most of the photosynthesizing, all of the photosynthesizing microbial plankton are mixotrophic. So that would include the cyanobacteria, that would include the diatoms. But now when we add phagotrophy as another type of heterotrophy on top of phototrophy and osmotrophy. So this is using particulate feed, so using food. So it involves grazing, predation. So it basically involves removal of an organism from the food web. Then we have then we don't have the cyanobacteria diatoms anymore because there's no evidence that these organisms can eat. But then we get a mixoplankton. So now we have a whole lot of different protist plankton, which are making food through phototrophy, and then also using osmotrophy and phagotrophy in their nutritional strategies. So hence, to make sure we were actually looking at the right community, we proposed a mixoplankton. Also, the difference between a mixotroph, which would not be doing phagotrophy, as a mixoplankton, is there would be a different impact on the trophic structure, the food web, because a mixoplankton will remove other organisms, whether it's another prey, a competitor, etc., by eating, whereas a mixotroph, which is doing only phototrophy, osmotrophy, is not doing that, is not removing anything through their mixotrophic ability. So from now on, we'll use the mixoplankton because we're looking at mixoplankton. And the next question was, okay, we've got mixoplankton. So how many types are there? Do we just have one type? Can we put them all into one type of mixoplankton? We've got phytoplankton, protozooplankton in the middle. Can we just have one mixoplankton? And in order to answer this question, we went back to the basics. So we started with the protists, and we tried to derive a functional grouping key. The first question we asked is, are these protist plankton capable of fixing carbon? So can it photosynthesize? If the answer is no, then we get our traditional protozooplankton. So these are the animal-like microbial plankton, which are only eating and not doing anything else. If the answer is yes, then we ask the question, okay, we've got a protist which is photosynthesizing, can it then eat? So can it phagocytose, or can it eat? If the answer is no, then we get the other end of our dichotomy, the plant-like end, the phytoplankton, which are only taking up nutrients and using carbon dioxide and sunlight energy, etc., to make food. So now we have got these are our the traditional plant-animal dichotomy. So when there's a no, then the next question is obviously we're still here. So there's a yes. And the yes is okay, so that means we now have an organism. We've got a mixoplankton, which is fixing carbon and eating. So this is where we were in the last slide. So now the question is, okay, how is it going to be fixing carbon? Has it got its own photosynthetic capability? Has it got its own chloroplast, which it can use to fix carbon? And if the answer is yes, then what we called it was a constitutive mixoplankton. So this is a schematic of a constitutive mixoplankton. So what we have is we've got a single cell. It's got its own photosynthetic factory or its own chloroplast, the capability, the genetic code to maintain its own chloroplasts, and it also eats alongside. So it's using different light energy and different nutrients to make food, and it's also eating. And in this we have So we have got various quite prominent examples. Here you can see we've got a mixoplankton, which is eating. So what it's done, it's basically stuck a peduncle like a straw, and it's sucking the food in. So you can see it's going into it. So essentially, so this one's now come in and stuck its own straw into. So there are lots of different ways they eat. Some will put a fishing net, some will stick a straw in. This one's engulfment. So what's this doing? It has captured the prey here. So you've got the mixoplankton predator, which has captured the prey, and it's gradually eating it. And you can see it starts with sucking a few things, and then gradually it's going to start and see if it can take the whole lot in one go. And it's nearly there. So there we go. That's all gone. So that's eaten. It's food. And this one is another one. So what this one has done, it's captured the prey, and again, it's going to engulf it. So can you imagine this is happening in the oceans with all the waves, etc. These little organisms are trying to capture these things. Here's another example. These are two different mixoplanktons. So this is a tripos and this is a Fragileidium. So what we've got here is a small one is attacking the big one. So we've got two of them, and then there's going to be, and they're literally engulfing. So there's another one and they're literally eating them up. So there you go. So three of them are eating it, crunching it up, and they're going to get to the end. They're going to fight over the last piece. This and nearly there, all gone. So clearly they want the last bit. There's something there which they don't like, so they've left a bit. They're still fighting, and there's a bit they don't like, so they've left it. So these are a few examples of constitutive mixoplankton. And the question then was, we wanted to see where can we find them? What can we find in the biogeography? And we looked at the different types of constitutive mixoplankton according to size. So in this one, on this axis, you can see the size. So it goes from less than 5 micron to over 20 micron. And these are the broad taxonomic groups. And what we can see, they're absolutely everywhere, just looking at it broadly. What we also found, there was a bias towards certain groups in the studies. So the fact these are white doesn't mean they don't exist. It means people haven't looked for them. The blank spaces indicate that you don't get any of these types in that size range. So there was a clear, in science research, there's been a clear bias towards studying certain types of constitutive mixoplankton. So back to our key. So we're back. So there was a yes. So obviously there's going to be a no. So now we have got mixoplankton, which is eating and is photosynthesizing, but not using its own chloroplasts. So what does it do? It has to get it. It's a quiet phototrophy. It has to get it. The question is, where does it get it from? Does it get it from specific prey, or can it get it from non-specific? So the question we asked was, is a quiet phototrophy from a specific prey? The answer is no. So then we get our generalist non-constitutive mixoplankton. So non-constitutive because they do not have the ability to make their own chloroplasts, mixoplankton because mixoplankton, generalist because they're taking generalist prey, generic prey to get their chloroplasts. So here we've got a schematic. We've got a single cell. We've got, in this instance, we've got phagotrophy, predation is coming through its digestive vacuoles. And then for phototrophy, what it's doing, it's taking in prey. It's going to keep the chloroplast or sometimes parts of literally just the plastids from it, kleptochloroplast, and they were going to use that to photosynthesize. So the thing with this is because they do not have any specific prey, they can steal their chloroplast from a lot of different prey. What they need to do is they need to keep topping up. They cannot look up. Their maintenance capability is it's not really there. So they need to keep topping up. So they are very much dependent on the prey being in the environment. Examples of these include the really beautiful ones. So we got the Lavoia and the Strombedium, and they're also very well. They look pretty, but they're also very colorful, and they're also the preferred food items for the mesozooplankton. So where can we find these generalist non-constitutive mixoplankton? Again, we did a biogeography study, and we can find it in every different systems across the world. Again, the white spaces means that people haven't looked at them. In essence, if you don't look, you won't know they're there. But we were quite surprised to see you can see them in oligotrophic systems, eutrophic systems from from equator through to poles. But these are not easy to grow in culture, so they're not very well studied. We don't know much about them. Back to the key. So we've got the generalist, obviously. The next thing is there's obviously Mixoplankton, which are specialists. They need to get their acquired phototrophy, to get their plastids, they need specific prey. So within this, then, when we looked at it again, we found there were two types, the plastidic and the endosymbiotic, and I'll get to each one of them separately. So we start with the plastidic. We've got the, these are needing specific prey. So therefore, for phototrophy, they need specific prey. So they have got a better maintaining capability. And then, of course, they've got the predation they're eating. So they will take in chloroplast from a special prey and keep it. Some of them can keep it for up to 3 months. And then they're also on the other side, they're also eating and digesting. The important thing which we found out, because originally when we started looking at it, the idea was that it was either doing one or the other was a top-up mechanism. But what we have found that it's all very much a synergistic. It needs both the phototrophy and the phagotrophy. So an example of this includes the Mesodinium rubrum, which is a very widespread, well-known ciliate, and it needs the plastids from this cryptophyte, very specific cryptophyte clade. So what it will do is it will take these teleolax in, and it will keep its plastids, and it also keeps some of the nuclear material, and it can go for about 3 months without needing a top-up. Then we have got another example of the plastidic SNCMs, are the Dinophysis. These are also very well-known species, bloom species. But what these need, they need the plastids from the teleolax. But because of the size, this is quite small for them to take in, they need to take it in via the mesodinium. So basically, the mesodinium does the stealing, and the Dinophysis then goes and does the robbery. So that's how, so it's quite an interesting complex when you look at them in the environment. And here's a lovely video by Mangyun Park, which shows, when it starts. I don't know, it won't start. Well, it's available on YouTube. What it shows is basically the Dinophysis, again, like the other ones we saw, the Amphidiniums, it sticks a straw in, and basically it sucks everything out from the mesodinium as well. So where can we find PSNCM? So we looked at the biogeography because they were, when I started, and the idea was these things are not important. So I really wanted to find out where these things are. So we did a biogeography study of this as well, and we found, again, they're absolutely everywhere. And this was really amazing to find them, you know, again, I think it's amazing to find them, all of them, everywhere, all the time. So the next one we have, the specialists, are the endosymbiotic ones. So what these do, these are even more fascinating. They don't steal anything. They keep the entire prey or as a symbiont. So their phototrophy, again, we've got the single cell, one cell, they're digesting, they're ingesting, prey, eating, but they will also take in prey or other photosynthetic organisms and keep a whole community inside to do photosynthesis. And there are some well-known examples in this group. They are the biogeochemically important forams and the Radiarians. So in this foram, you can see this is a central capsule. These are its spicules, and all these little dots are the symbionts. So basically, there's sort of a gelatinous external cytoplasm, and in there they've got what they do. It's when it's light during the daytime, they will send these symbionts out. So they'll be out there, and then as it grows dark, they bring them in and keep it in here. This is another one, an acantharian, and again, you can see this inside. These are the dots are the chloroplasts again. Sorry, the symbionts, and again during daytime they will send them out, and then at night when it's dark they bring them back in. So these are the endosymbiotic ones, and there's an absolutely amazing, and I really hope this plays. I can find this maybe. Yes, this is an amazing video by Professor Spiro so here we have got a foram. This is an Artemia. So an Artemia is, or known as a sea monkey. These are used in aquaculture for fish food. So you can see how big this is, because this is about 1 millimeter long. So you can see how big the foram is. So what it's done, it's got a spicules, it's captured the Artemia, and as you'll see, it's gradually pulling it in towards its central capsule. So so it's gradually pulling it in, and then what's nice is what Professor Spiro does, he then zooms at the other end, and we'll soon see the symbionts. And there we go, we can see all the symbionts now. So you can see the little dots there, and those are the symbionts of the foram. So it's got a central capsule symbionts, and it's now gradually getting its prey in. It will put it in and digest it. So that's food. So where can we find them? This was a surprise, because the original idea is these, what we thought these would be present in oligotrophic systems, so in the middle of the oceans, etc. But when we actually did the maps, this is what we found. They were absolutely everywhere, and from the poles, again, from the equator to the poles, and oligotrophic to eutrophic systems. So that's the ESNCMs. So what we found with our key and looking at the biogeography, that the Protist functional groups were a bit more than just the traditional photosynthesizing plant-like phytoplankton and the consumer animal-like protozooplankton. There's actually 4 more, and this is very broad classification. If you talk to taxonomists or talk to molecular people, they will say there are more than this. So that's the so we've got 6 functional groups. So it's going back to the title of the talk is, what are they? Are they mini monsters because they're doing all these things, or are they perfect beasts? So we'll start with the iconic Emiliania Huxley, or called EHux. So it's a coccolithophore, where we can find these are made of calcium carbonate or chalk. It's a coccoliths in a coccolithophore, and it has always been a traditional phytoplankton. And we can even find mention of these in the literature, where, for example, in Jules Verne's 20,000 Leagues Under the Sea, where he talks about the Nautilus sailing into the Milky Sea. And this Milky Sea we can see from with satellite from space. So this is the coast of Cornwall, and you can see in the sea the coccolithophore blooms. The other place we can see evidence of coccolithophore existence in the past are the White Cliffs of Dover, where the white comes from these calcium carbonate shells of the coccolithophores. This has always been an iconic phytoplankton, but very recently it's been found to actually be capable of ingesting bacteria. So we now know that these are mixoplankton, so it's photosynthesizing and also eating. In a healthy ecosystem, mixoplankton like the EHux, then the tripos, which you saw was being devoured in one of the videos, the cryptophytes, which gives chloroplast to the mesodinium, and the mesodinium as well. They support the higher trophic levels. So they are food for copepods, for krills, which then support fisheries, whales, dolphins, etc., all the bigger enigmatic megafauna. However, in an unhealthy ecosystem, what we can get are blooms, monoculture blooms of these. So single organisms can happen and occur in blooms, and that's and what happens then, we got problems. We get fish kills. In different arenas. We get aquaculture farms, etc., being shut down, and a lot of this is because of the changes in the nitrogen, phosphorus content in the water. So fertilizers have been considered as one of the things, and we also get affects us and indirectly by causing different types of problems with the shellfish as food for us. So this is an example of a Calodinium, which is a single-celled constitutive mixoplankton, and this is a copepod, which generally would be eating phytoplankton and then being eaten by fish. So you can see the Calodinium are attacking them. What they're going to do is gradually bite bits off, and then what we've got also seen videos of where the Calodinium will immobilize this this copepod and also an adult copepod and kills it and eats it through. And we've seen that same with the nematodes, so worms and also different sea slug larvae, etc. This is the worm one. Yeah, I've got one. So let's see. There we go. So it's basically attacking the nematode and it's literally eating chunks of it. So basically, going back to the question, mini monster, nature, perfect beast, it depends on us really, how we look after the environment, what we get. So what does it mean? So we've got this new community, we've proposed a new term. What does it really mean in terms of the marine ecology? Does it matter? And why does it matter? So I do mathematical modeling, and one of the things I wanted to see is what is the impact of putting mixoplankton in mathematical models of food web models of marine ecosystems. So before we go there, these are some of the examples of the climate change models which are used, global climate change models used for the IPCC type projections. And when we look at the plankton community here, we can see they've got the nutrients. So they've got nitrogen, phosphorus, Si, silica, iron. And then when we look at the P as a phytoplankton, Z as a zooplankton, we can see there are some have 1, some have 3, some have 2 zooplankton, but there are no mixoplankton anywhere. Looking at the climate change models, if we look at just the plankton, even without the mixoplankton, when we look at it, we've got the cyanobacteria, the phytoplankton, the zooplankton, the bacteria. It's pretty complex. But when we put the mixoplankton, it's even more complex. And so this is all which is missed out in these climate change models. But the question then is, does it really matter? Can we still get away with 1 or 2 boxes representing phytoplankton, zooplankton, and just say, okay, a certain percentage is going to be mixotrophic versus not? So to do that, I looked at 3 different food webs. So I looked at a traditional food web, so where we've got a phytoplankton, we've got this protozooplankton. So the phytoplankton is using light, carbon dioxide, etc., to make food. The microzooplankton's eating the phytoplankton, and then we've got the bacteria, which are the regenerators or the decomposers in the system. Then in the next scenario, I replaced this the protozooplankton with a constitutive mixoplankton. So these are our tripods, the Ahux, the Calodinium, Alexandrium, etc. So they fall within the constitutive bunch. And in the scenario 3, I replaced the protozooplankton with the non-constitutive mixoplankton. So these are our plastidic cilia, so pretty ones, to see how it works. And then I ran these 3 simulators to see what happens under different conditions. So we can look, I'm going to show only one set here. So we've got the traditional, and here we have this on the x-axis, we've got days, time, and the y-axis is biomass. So it's in carbons per liter. So we find that over time, gradually, the phytoplankton bloom happens, and then the microzooplankton, the protozooplankton follows it, and the bacteria is in the background. So that's a traditional thing. That's what you'd expect, a nice clean bloom of the spring bloom, and then the zooplankton. Now that, if you got that, if I had that 10 years ago when I started my PhD, I would have been very happy with that. But now when we put the constitutive mixoplankton in the same, again, we've got days, so it's up to 30 days we've done, because usually these blooms would be about 30 days. Within 30 days, you get a change. And then you've got a biomass here again, so the same scale. And what we see is very different. We see that when we have this system, the constitutive mixoplankton absolutely take over. So they're there, and this would be a situation where we can get a harmful bloom situation. And in the third scenario, when we replace it with the non-constitutives, or the ones which have to acquire phototrophy by stealing plastids from their prey, we get the similar bloom like the first one, the traditional, except we get a lot more biomass for the non-constitutive mixoplankton compared to when you have the phytoplankton. So the amount of carbon they're fixing is going to be different for the different scenarios. So in essence, the take-home message for me from this was that what matters is how we configure our food webs. If we do not consider the mixoplankton, then there's a chance we are not going to get the correct dynamics. But the next step was, this was a simple one, to put it in a more complex scenario. So what for this, what Susanna, one of our PhD students, what she did, she took the ERSEM model, which is a European Regional Seas Ecosystem Model run out of Plymouth Marine Lab. And there she took the plankton food web, which is on this axis now, we've got the sizes. So we're going from small pico to meso big here, small to large. And she replaced all the compartments which are known to have mixoplankton. So she replaced them with mixoplankton functional groups. And then she did lots of different experiments. And what she found is that, again, if we consider mixoplankton versus if we don't consider mixoplankton, there's a massive difference in the biomass. So here we've got the different types, and we've got the biomass on the axis, the x-axis. I think I'm nearly done now. So basically we found what my question was at the end of my PhD when I started looking at mixotropes. I wanted to see if they were important, more than I expected to find. Okay, they were important, but I did not expect to be proposing a new term as a community of Protist plankton. So to summarize, in 100 years of marine research, we have got countless textbooks, BScs, MScs, PhDs, grants, etc. We've got climate change models projecting future life on Earth. We've got 2 series of Blue is the Blue Planet, and all these missed this. Yet 50% of the oxygen that we're breathing in now, breathing in every day, it comes from the microbial plankton, the organisms that most people don't even know exist. So what are we doing about it? We have got a grant which is running at the moment. It's a Marie Skłodowska-Curie ITN, which employs 11 early stage researchers. So we are exploring different techniques of how to study mixoplankton. We've got, and it's been recently highlighted as a success story by the European Commission, which is nice. We've also got in Cardiff a Circumry grant, which is setting up a mixoplankton research hub, again looking at different mixoplankton, importance of plankton in marine systems. And what we have got so far, we are revising research methods. So if you think of most marine plankton methods, they are based on either the plankton photosynthesizing or eating. So it's either looking at something which is making food or something which is eating. So the techniques, what we found in Mixotin, most of the techniques are not suitable as it stands because it has to do both. So we're revising these research methods, which obviously changing, and therefore we're changing scientific outlook. We're currently building the first ever mixoplankton database. So this is to put all the microbial plankton we know of, which are mixoplanktonic, looking at the sizes, looking at what they eat, etc. So we're doing that. We are also engaging with different media. So I've written conversation articles. I was asked to write an article for the Scientific American, which was really nice because it picked it up for one of the special issues, and Mixoplankton has been considered, along with black holes, etc., as one of the most important discoveries out of 13 discoveries that could change everything. We're also, because one of the important things is, and especially in this decade of the ocean, is having a healthy ocean, a sustainable ocean. So it's very important, at least I believe it's very important, that whatever we find in our research has to have a link into applications in real life. So one of the things is with all these the potential bloom problems and etc. is on the policy side, the water management. So we're trying to work with policymakers to find ways to monitor mixoplankton. We have so far at the moment we have recently we have developed a key. From the key I just talked, we have actually got a key which is specifically targeting HABs, and we're looking at the IOC UNESCO HAB list for that. We have looked at that. We're also, the other important thing is, of course, education. People don't know about mixotropes, and if they don't know about mixotropes, the reason is probably because scientists haven't talked about them enough. So that's very important. So we're writing different, again, articles which are for children. I'm a STEM ambassador. I also go to schools, etc., to talk about mixotropes models, etc. And another thing which we have done out of Mixotin is we have developed a simple model which we have used for lots of different outreach activities to teach children, to teach high school students, or pupils from junior school to high school, and even undergraduates, of the importance of our environment, impact of what we do, eutrophication, etc., on mixoplankton. So there's a lot going on, a lot to do. It's a lot of stuff, challenges, fun, etc. So with that, I shall leave you. Thank you very much for your attention. If you want to find out more, you can follow us on Twitter, Facebook pages, and on our webpage, which is mixotrope.org. So thank you.
Thank you very much, Aditee. That was absolutely fantastic.
Shall I unshare now?
Sure. Or you could leave it if you'd like, in case people want to write it down. We've had a couple of questions come in. One of which is, someone's asked, when there are bloom events, are these only of phytoplankton, which can survive on sunlight and dissolve nutrients, or can they happen with mixoplankton, which rely on other species to survive?
One of the things we're looking at is that IOC UNESCO list. So yes, the blooms, when you get the cyanobacteria bloom, yes, they are all phytoplankton because they are mixotrophic phytoplankton because they're doing osmotrophy, phototrophy. But when you get the Protist blooms, so the Calodinium, Alexandrium, Primnesium, all the ones I showed, they're all constituted mixoplankton. So they're eating and they're also photosynthesizing.
So mixotrophs can cause blooms as well.
Oh, they are all these blooms are from Dinophysis. Most of the HAB species which are in the IOC list, once you take out the cyanobacteria, they're all mixoplankton. So yeah.
Yeah, so someone else has asked, are there certain species of mixotrophs which are more efficient or capable of nitrogen fixation?
Nitrogen fixation is mainly going to be cyanobacteria, so that's going to be, yeah, so the ones I've been looking at
are the ones, so I guess that begs the question is, are the ones that have symbioses with cyanobacteria, the symbiotic organism is a cyanobacterium, are those capable of nitrogen fixation as a kind of symbiosis?
You know what, that is a question which I've been looking at, and because we can't grow those ones, the endosymbiotic ones, what the question which I haven't found the answer to is whether they can take up nitrate, or because unless they can take up, have got the transporters, and because if they've started as a protozooplankton, when we look at things like the lab rat type protozooplankton like Oxyrus, they don't have transporters to take up any nitrate or nitrite. So if it goes with that, with the endosymbiotic ones, and they haven't evolved to have any transporters, so they will not be fixing nitrogen. But the cyanobacteria symbionts, what they would be getting will be the ammonia from the from the excretion. From the excretion. So when from the digestion process is going to release the ammonia, which then the cyanobacteria will be able to get. But we don't know, because in the database I've got a list saying can they take up nitrate, and all the ones with these I have got as unknown.
Question mark.
Yeah, literally, yes, that's what it's. So we don't know.
Because that kind of begs the question of could you use these to perhaps kind of alleviate pollution effects in water if they're able to break down nitrogen?
Well, the whole problem is you'll have the blooms is because there's a mismatch of the stoichiometry. So the ratio of nitrogen to phosphorus is not as it should be for a balanced. So for pollution, it will, depending on if you've got too much nitrate and not enough phosphorus, it depends on which is going to turn up, and then you will end up with toxins. So it's not a simple solution that you grow these and that will take up the nitrogen. Would
be nice, wouldn't it?
It would be nice, but it would release other things.
Peter says, wonderful talk, and have mixoplankton been found in freshwater as well? So they're in the ocean, but are they in freshwater?
As far as I know, yes, but it's not something I have looked at because of my research has mainly been marine. But there are the original definition, the descriptions. There was Dan Stoker who did the Mixotroph conceptual model, and there's Harriet Jones who did one for the freshwater system. So it's very similar. They're both the concepts which evolved in the two different systems. There's also work done by Bob Sanders. He also worked both on marine and freshwater. So yes, there's Diane O'Brien is the one which I know of, but I can't say how much of the the Protist plankton in freshwater systems are mixoplankton.
Work to do, isn't it?
Yes.
And somebody's asked about the graphs you showed, the 3 graphs with the models. And he says that the third model showed the microzooplankton increasing before the mixoplankton. How does that work, and what were they eating?
They were eating the bacteria, because the microzooplankton can eat the bacteria. So it's because the traditional one is based on the microbial loop, where you've got the and I noticed that I think I don't have that linkage clearly, but the microzooplankton would be eating phytoplankton and bacteria. So basically, what the traditional paradigm, you will have the phytoplankton making food, the microzooplankton then eating the phytoplankton, bacteria doing the decomposition. Then the microbial loop, which was in the 1970s, 80s, the paradigm there was basically the bacteria are also being eaten by the microzooplankton. So suddenly it wasn't the microzooplankton and bacteria competing for organics. The microzooplankton was eating the bacteria. And now what we have found is now, because the phytoplankton has now become a constitutive mixoplankton, it's now eating the bacteria. So in a sense, the constitutive mixoplankton, so what would have been the traditional phytoplankton is, you would think it's going to be, not you would think what it's going to be sort of eating what would have been its competitor for nutrients. But going back to the question, how it would have increased, it would have increased by eating the bacteria, but not. So that's why, yeah.
Someone has said they're having difficulties with the use of the prefix phyto, which means plant, plant-like, or plant-related for this group of related organisms. The only similarity plants share with phytoplankton or plant-like or vice versa is the fact that these organisms can use photosynthesis. Of course, you could argue or ask the question, what is the definition of a plant? Any thoughts?
No, I haven't looked at the definition of plants, though. Though I should have, as a stuff, as an original botanist. But I have looked a lot at the definition of mixotropes, and from there again, it's the same thing as with the problem of, where it's come from and looking at the origins. I don't know. You'd probably be better equipped to answer the plant question. From mixotrope things, a lot of the words like osmotrophy, chemotrophy, auxotrophy, it comes from microbiology rather than botany or protist science. I don't know. It's a
Well, I think it probably goes back to that kind of big division, which was it's either a plant, a rock, it's an animal, a plant, or a mineral, which was, of course, Linnaeus's three kind of realms of nature was mineral, plant, and animal. And so, you know, sticking those Latin names on it gives it meaning, perhaps in an old sense, but perhaps we have to think a little bit more about the boundaries, as you said, very, very eloquently, is those boundaries are a bit more fluid than we thought. They're not so clear. And I think that's becoming true really about a lot of things in science, is that the things we thought of as being discrete categories actually are very leaky.
Because it's higher plants, again, is going to be very different from things like seaweeds, which form in the plant kingdom.
Some do and some don't.
Yeah, that's true. Yeah.
So we, you know, and of course, fungi used to be considered to be plants as well.
Yes, of course. But yeah, they've got their own kingdom.
They've got their own. They're their own quite amazing thing.
Yeah.
Someone has also asked: is there any evidence the release signals to that they release signals to communicate with each other? These mixoplankton who are hunting in packs, or is it just random chance when several or one species attack the same prey item?
Well, it's there is something called allelopathy, which is known. People know about it. So that's a chemical signals which stops the other from working. So or will do, like one of the plankton will take up, bind up all the vitamins to prevent its competitors from taking it. So that's not exactly a chemical signal.
Kind of the opposite, isn't it?
Yeah, as the opposite of how to prevent. Whether they are talking to each other, don't know, but it's all happening. The blooms will usually happen because there's a change. There's something in the environment will trigger it. If we knew the answers, we'd stop it. We know what probably is, but so no, I don't know the answer to that. I don't know. It's a good question.
People didn't used to think that plants didn't communicate with one another, but now we know they do. Higher plants. So
yeah,
perhaps mixoplankton.
I suspect there will be something. There's also some of them will release this toxin which bursts other, competitors or prey items. And the question there is, how is it not? It must have evolved that its own, wall or membrane is not lysing, because all the others are lysing. So we can't grow these things. For example, primnesium, it will cause, serious harm to fish, gills, etc. And when we try to do experiments in the lab, you cannot even put anything with it without, but somehow it can grow, form blooms, and it's not lysing its own cells. So who knows?
Amazing. So what someone's asked, what are the implications of extra biomass so low in the food chain being not taken into account in these scenarios? So what are the real implications of not, taking mixoplankton into account in these climate models?
Well, basically you can get the wrong type of plankton. So if you're looking at, for example, if you had a traditional dichotomy of phytoplankton making food, zooplankton eating, and then the fish eating, etc., and you missed out on the bloom, you can have an entire collapse of a system. So one of the things which has happened in it's a recent paper showing in the middle of the Atlantic, they're finding that gradually there's been more and more cyanobacteria blooms, which is then affecting of what's going to eat those and what's going to go up. Likewise, of the coasts in Southeast Asia, they have got a change. They've literally got a change from blooms of diatoms to blooms of dinoflagellates. As a result, the whole ecosystems changing from, it would have been diatom, copepod fish, it's gone to dinoflagellate, jellies, sea jellies, salps, etc. So it's changes in, it changes the whole thing. So if you don't know what is out there, it may not make any odds. If in your particular system, if you don't put it in, it might not make odds, but unless you put it in and you show that it isn't making any odds, you won't know. And we know enough to know that they are there. Most of them now, other than the diatoms, most of the Protist plankton species, other than diatoms, have some level of mixoplanktonic species in them. So therefore, if you don't account for them, you will not necessarily know what's going to happen in the future or now.
Yeah, so somebody's somebody's suggested to use phototroph and heterotroph, but then there's mixotroph as well. There's lots of words that people use. Somebody else has asked, is there any evidence of environmental contamination in these systems due to microplastics? So is there any evidence of uptake of microplastics by these microorganisms?
Well, there were experiments done way back in the 1980s by Dave Karen and Goldman. There are some iconic papers by Karen Goldman papers, and they worked with the Protist plankton, tried to feed it latex beads, because they were trying to at that time, they were trying to get grazing rates. They were not thinking in plastics terms, but it was really interesting because they would recognize it. So they would take not Dave and his colleague, but the
plankton,
the plankton, the microzooplankton or protozooplankton would recognize, no, it's not food. So after, it would take in one or two, but after that it would reject it. So there's plenty of evidence that they rejected. They will not eat. They're not stupid. They will not eat plastics.
Well, I think that's evidence indeed. Someone else has asked that from your distribution models, the maps you showed,
the biogeography, yeah.
Was the data only from C column samples, or have you modeled individual species distributions, say for example?
No, those are field data. So those are from OBIS and Susanna. So Susanna went through a whole load of different databases. So she looked through all field data. So those are data from.
So it's a mix of sources.
Yeah. So there's all databases, Copypod, OBIS. There's a list. If you look at the papers, there's a whole list of. So literally, she went through every single database. She's designed a code to extract them, and she I can't remember exactly how many, but there's tons and tons. So they're not. Sorry.
Clearly, people need to do a bit more sampling in some parts of the world.
Oh yes, absolutely. It's not just that. It's also if you think about it, when we go sampling on a field trip or something, you're going to cruise 10 stations, and because you have these blooms in patches, if you put your CTD or if you put your Niskin bottle in this area and a bloom is here, you missed it. You won't know. Plus the other thing is, if you're looking, and the other problem is people haven't been looking for these. So they have been looking for either a phytoplankton or phototroph or a microzooplankton, protozooplankton or heterotroph. and because you're not looking for a mixoplankton, you won't. Because one of the interesting things which I found out from Mike Zubkov, who found the flagellates the first time, this was in 2004, 2006, as a Zubkov, Tarrant and papers, and they reported mixotrophy in the flagellate bunch in the Atlantic. And what he said, he went looking for the plane again, the autotrophic and the heterotrophic, and he couldn't find one. So then he noticed that he was literally taking in one bacteria in a day or something. So if you missed it, eating the one bacteria, you missed it. You don't know what's happening.
But people have sampled Emiliania Huxley for ages. I mean, no, that's hugely widely distributed.
Yeah, but they haven't looked for it as whether it's eating. They just assume
they haven't looked at it.
Plankton.
I see what you mean. Yeah.
They've just been looking at, so there's a whole lot, but it doesn't say. So the thing is, what bacteria were around, and if anybody's bothered looking at the bacteria.
So it's a question of not just sampling, but also sampling and sampling behavior as well.
Yeah, sampling, and that's the main, and that is what we came across in Mixo10, is the fact it wasn't just sampling, it was also how we are sampling, and therefore whether we are sampling the right things or we're capturing it, because it has to be. So, for example, the continuous plankton recorder, it goes across the world collecting samples, but anything, the microbial plankton, other than the tripos, the one with the horns, which was eaten up by the tree, other than that, they cannot capture anything because everything just turns into a gelatinous mush. So you have no idea. So
why does it turn into a gelatinous mush?
Because because that's what it is. So it's not got a strong structure to capture. So
how do you sample differently? So how do you sample that's a different way to the way other people sample?
So if you're sampling, for example, the endosymbiotic ones, it has to be plankton nets, it has to be just below, and you have to gently pull it with the boat. So and if it's the most of the ones, the constitutive ones, they are about nano size. So those you can just put Niskin bottles and get them from different depths. So those are fine. But the thing is, microscopy, you have to look and make sure you're pulling out the right one. So that's the other problem, is people don't necessarily know the plankton either. So taxonomists. Good taxonomists are not.
Yeah, just chucking it in a jar isn't good enough.
No, and also how you, if you're going to fix it, how you fix it, because if you're fixing with, for example, formaldehyde, they might just burst, you might not get it. So it's also knowing, Lugo. We have got, for isolating and setting up, establishing cultures, we have one of the things which we have got from Mixotrophy, and we're waiting, we've submitted to the commission, we're waiting for the rubber stamping. Is how to isolate and set up Mixoplankton cultures. So anybody should be able to pick it up. We're trialing it with MixoHub. So Angela, who's my postdoc, she's been trialing, so we know it works. She has managed to set out 20 cultures, which is absolutely amazing.
That's fantastic.
So
something that's basically unculturable. That's terrific.
Yeah. So that's.
Oh, well, I don't see any more open questions, and you've answered loads, and I thank you so much. And that's open for me. That's opened an incredibly new and exciting world, and thinking about how you sample for something and how you study it and how you can, and it also, I think, for anyone who's listening will realize there's a whole lot out there still to learn. There's a huge amount still to learn, which I think is one of the most exciting things about being a biologist now, or being a scientist, is we discover things all the time that show us how much more there is to learn. So thank you so much for tonight's lecture, Aditee, and
yeah, thank you for having me.
The best.
Can I ask a question, since I've got the Linnean Society here?
Yes.
Carnivorous plants.
Mm-hmm.
Do you know what they are doing? So when they're taking the fly or the insect, when it's digesting it, what exactly is it absorbing versus what?
Nitrogen.
So it's only absorbing nitrogen.
No, it absorbs everything. It absorbs all the nutrients that.
Right. So
I'm no expert on carnivorous plants, though. I'm sure somebody else knows much more than I do about carnivorous plants. But carnivorous plants often grow in sort of nitrogen poor areas.
Yeah, I know, and because I know if we put water, rainwater, because otherwise it's not going to just normal water.
There's on our YouTube channel, on the Linnean Society's YouTube channel, about, I don't know, 2 or 3 months ago, maybe 4 months ago, we had someone give a most extraordinary talk about pitcher plants capturing.
Oh, okay, I'll have a look.
So have a look on the YouTube channel.
I will, thank you. Yeah,
we had amazing kind of electrodes and measured, kind of how things slip, and it was quite extraordinary. I can't for the life of me remember what his name is now. That's very embarrassing, but it's on the YouTube channel.
Thank you. I'll do that. But thank you so much for having me.
Thank you so much for that amazing lecture, and thank you as well to the Royal Society of Biology, the South Wales branch, and for hosting this, and Cardiff University as well, and to Diane, who started this entire thing off, and long may it last. Thanks a lot.
Indeed.